Well, let me begin by introducing our first speaker, who is Dr. Robert Edwards. He's a professor of neurology at UCSF. He received his MD from Johns Hopkins University many years ago, and then trained in clinical neurology at UCSF, and also did a training as a fellow in biochemistry and molecular biology. He's been at UCF for many, many years now. Many of you may have heard him talk at previous conferences of this sort. He's received many honors and awards over the years, including international prizes, but I'm particularly pleased to tell you that he was elected just a few weeks ago to be a member of the National Academy of Sciences. Let me just tell you what that means. The National Academy is a collection of about 1,800 very eminent scientists from the United States, and perhaps about 400 from other parts of the world. Of those 2,200, approximately 10% have gone on to win Nobel Prizes. So this is a very august body. We're very, very proud at UCSF that Robert Edwards has now joined those ranks. Anyway, so Robert's going to talk to you today about the cause of Parkinson's. Robert.
Thank you, Michael. Thank you for coming out today. So yes, it's a little bit of a tall order, the cause of Parkinson's disease. So as we all know, we have treatments for the symptoms of Parkinson's disease. That's the good news for this disease. Unlike so many others, we do actually have something to give patients that make their lives better. Of course, dopamine replacement with Cinemed, dopamine agonists, and eventually, potentially deep brain stimulation, all of which are pretty successful. But the problem is that, as we also know, the disease continues to progress. It's been progressing before the symptoms even appear, and it continues to progress afterwards. And we really don't have anything to address the underlying degeneration, which is really what causes the disease. So what are our clues to what causes this disease? Basically, in the last 10, 15 years, there have been identification of a number of genes, mutations in which cause Parkinson's. So, there are several genes, mutations in which cause Parkinson's disease. And several of them cause what are called autosomal recessive forms of the disorder, which are due to the loss of particular genes. That's not what causes typical Parkinson's disease. Usually those people get Parkinson's in their, okay. Excellent. Okay, thank you. Okay, here we go. So, this is. I thought this is. I think I know what. There we go. Okay. There we go. Okay, so there are mutations in some of these genes called Parkin and PINK1. They cause autosomal recessive forms of Parkinson's disease. Those are Parkinson's disease that begins usually in the teens. It's not like what most of us have. So I'm not going to really be talking about those today, although a lot has been learned about Parkinson's disease from these very rare disorders, particularly the autosomal recessive forms, and their role in maintenance of mitochondrial function. What I'm going to talk about today, those don't usually account for idiopathic Parkinson's disease or adult onset Parkinson's. So what I'm going to talk about today are the ones in red here. These are the inherited forms of Parkinson's disease due to mutations in two genes, alpha-synuclein and a gene called LRRK2. Now, just before I begin this, the mutations in this cause a very small fraction of all the Parkinson's disease out there. But the point is, because we know about these genes, we understand something about what's going on. So we can learn from them about what might be going on in everybody else. So, so that's basically the idea. These are a clue. So mutations of synuclein, mutations of LRRK2. LRRK2 mutations are actually fairly common, relatively speaking, still a small percent of all patients, particularly in Ashkenazi populations or Semitic populations. And synuclein mutations are actually extremely rare causes of Parkinson's disease. So fine, I can say mutations in these genes cause Parkinson's disease. But what does that mean? I mean, how does a mutation in those genes, how does it actually produce Parkinson's disease? We can find the gene, the mutation. So the question is, how do these work? Now, synuclein, again, the mutations that cause this are extremely rare. So what I'm going to talk about is synuclein today. And why am I going to talk about that? It's very simple. Even though it's so rare, basically, there are several reasons. So this is the protein alpha-synuclein. It's actually a small protein, 140 amino acids. It's just a little protein that our brain, all of our cells in our body make proteins. This is actually a little one. It looks kind of innocuous. Mutations in it cause Parkinson's disease. I'll talk a little bit about the structure of the protein later. But actually, what causes a particularly severe form of Parkinson's disease is just an extra copy or two of the normal gene. So we each have on each chromosome one gene, and these people, some of these families, they have on one chromosome 2 genes, or 3 genes for synuclein. So instead of the usual 2 that most of us have, they have 4 or 3. They have a little bit more. The ones with 4 copies of the gene get extremely severe Parkinson's disease starting in their teens. Everything's accelerated. It's the worst of the mutations in synuclein in terms of causing the disease. Okay, so all of this is extremely rare, but why do we care about this? This is why this is important. First of all, there have been some genetic studies done in the general population of Parkinson's disease. And it seems like even in people with just regular idiopathic Parkinson's disease, there's something about this gene that predisposes them to getting that disease. It's not as obvious as some of these mutations found before, but there's something going on there that implicates this as almost number 1 in everybody. Okay. Number 2, and this is the main thing, since the identification of mutations in synuclein by Bob Nussbaum, who was then at NIH, and subsequently moved to UCSF several years ago, it's been found that this protein accumulates in the brain of everybody with Parkinson's disease. So, what the mutations tell us is this gene causes Parkinson's disease, and what the pathology, looking at the brains of patients, tells us is that this is responsible for basically all forms, most idiopathic forms of Parkinson's disease. So the mutations are rare, but the protein accumulates in everybody. So we really, just to emphasize the main points here, we don't even think so much about Parkinson's disease as a disease of dopamine neurons anymore, although it is a disease of dopamine neurons. But we find synuclein all over the brain of patients. This disease is a synucleinopathy. It's a disorder of synuclein, more than a specific disorder of dopamine neurons, although it certainly does affect. So this is just showing what we used to call the Lewy body. And you can see this is stained with antibody to synuclein. You see these clumps, these aggregates of synuclein here. And usually in the past, before mutations of synuclein was found, people would use other stains to look at the brain of Parkinson's patients. They'd find a little clump here, a little clump there. But when they used antibodies to synuclein, they saw them everywhere. What we saw before was the tip of the iceberg. This is the vast extent of the problem. So, just to give you an example, these are just some other brain regions. The brown stuff here is deposits of synuclein. These are not in the substantia nigra neurons or the midbrain dopamine neurons. These are other neurons. These are vagal neurons, RAF-A neurons that make serotonin. These are cells that make norepinephrine. These are other cells that make acetylcholine. Basically, you find synuclein in many different places in the brain, in everybody, basically, with idiopathic Parkinson's. So, there's some German neuropathologist at Brock's who actually did some staging of Parkinson's disease. And actually, the earliest stages are over here, shown over here. The earliest pathology in Parkinson's appears in that vagal nucleus, the dorsal motor nucleus of the vagus. And then as it progresses, this is just by looking at the brains of many patients who died at different stages of the disease. And then as it progresses, it moves into some other places, and then about sort of at stage 3 in their pathway here, it gets to the substantia nigra. So the disease has been going on for long before that. And then after that, it actually spreads from substantia nigra, or it progresses into other parts of the brain in the cortex. And this just shows the substantia nigra is actually down here, and this is pathology in the temporal lobe, in the cortex. This is synuclein pathology. So it's spreading in many different places in the brain where people didn't know that it was even occurring before. So, and this actually explains many of the so-called non-motor symptoms of Parkinson's disease. Some of them may be actually due to loss of dopamine neurons, but many of them are due to the loss or the pathology in these other cells. So constipation, the sleep disorder, REM behavior disorder, where people act out their dreams. Many of you may be familiar with that. Depression, which can often precede Parkinson's disease. Cognitive problems, autonomic problems. All of these are due to synuclein accumulating probably in more than just the dopamine neurons, in many other cells. So, okay, so the whole point of this is to illustrate how significant the synuclein pathology is. But again, the genetics says it causes the disease. It's not just a byproduct of the disease. It causes it, and it accumulates in all the brain. So it's not even just Parkinson's disease. It gets even more. So Parkinson's disease is clearly a synucleinopathy. Lewy body dementia is basically a variant of Parkinson's disease, where synuclein starts to accumulate in more than just the usual places in Parkinson's disease. It accumulates in the cortex and amygdala more prominently than in Parkinson's disease. So this is a pretty common form of dementia. There's also multiple system atrophy, which is a form of Parkinsonism, where the synuclein actually accumulates not in the neurons so much as in certain types of glial cells. And this is a case where I would say there's a pretty strong case for what's called the prion hypothesis, which is the protein, the abnormal protein can spread from one cell to another. I'd say multiple system atrophy is probably an example of that. Worked on by my colleague here, Stan Krusner, who developed this whole idea that proteins could spread across disease. Okay, so I want to get across a few key points here. Okay, so normal and abnormal. This is like, if I could get something across, this would be it. So synuclein is a normal protein. We make it, everybody makes it, and our brains actually have a lot of this protein, everybody. And it's normal. I mean, so the question is, in Parkinson's disease, is the synuclein disturbance affecting the normal function of the protein? Or is it affecting something that the protein starts to do when it goes bad? Okay? So there's the so-called abnormal function. And the first thing people think of when they think about abnormal function of a protein like synuclein, because it aggregates the disease, is that its abnormal function is this aggregation. It starts to accumulate, and that's bad. Okay? So there's a lot of attention been placed on the aggregation of synuclein, which it clearly does. All those pictures I just showed you is aggregated synuclein. It's not the normal stuff, it's the stuff that's all clumped up. And also the toxicity of synuclein. And so these are so-called abnormal functions, the bad things that synuclein does. But it just is very important to keep in mind this is a normal protein. We all have it. So it must be doing something that's good for us, too. This is extremely important. This is a lot of what my lab works on. Because I think this is the key to understanding. Again, I should have introduced at the beginning, I think, a lot of times when patients come and I see patients, and the one thing that they rarely ask me is, if you're there to confirm a diagnosis or make a diagnosis of Parkinson's, is they want to know, why me? Why am I getting this disease? And so that is the key question that we're trying to address. And understanding the normal function of this protein and its abnormal function is the key to this. So, a lot of people have studied these processes of aggregation and toxicity. They take synuclein, you can make it. You can make bucket loads amounts of it and dump it on cells and do this and do that with it and look at its properties. And this has been done by many different labs, trying to understand primarily this aggregation. But what my lab has mostly been interested in is more related to the normal function. What does it do in cells? I mean, that's where it is in our bodies. It's not just floating around in our bloodstream. It's in our neurons where it does something good, normally, or bad. Okay? The reason why we're particularly interested in the normal function of this protein is extremely simple. If you want to do something about any disease, your best bet is to do something early. Okay? And we know in this disease that eventually the protein aggregates, bad things happen, people develop symptoms, patients develop symptoms. But we know that before all that happened, that protein was doing something normally in the body. So we know that the normal function clearly precedes the abnormal function. And if we want to understand the first things that happen in the disease, we have to know the normal function so we can understand the transition to the abnormal function. And the second thing, as I just alluded to, is if we want to do something about this disease, it's much better to do something in the first step than in the subsequent step. In the subsequent step, you'll be chasing after lots of different things. If you hit the first step, do one thing, and you may be able to prevent or arrest the disease. So that is the goal. That's why we study the normal function of the protein, and we want to understand what it does in real cells. This is the mystery. Nobody really understands what it does normally. Okay, so many proteins that cause degenerative diseases like Alzheimer's, Parkinson's, the other proteins that cause these other diseases like tau, and A-beta peptide, and Alzheimer's disease, we don't know. They're in many different parts of the cell. Where they're located doesn't tell us much about what they do, and there's huge arguments about what those proteins do too. So synuclein is just like the others in Alzheimer's disease, like A-beta peptide and tau. Nobody knows what they do. Nobody knows what synuclein does. But we have one huge clue with synuclein. It's the advantage for Parkinson's disease, it's in the nerve terminal. What is the nerve terminal? So the nerve terminal this is a synapse. So these are two synapses here, formed onto the single, what we call postsynaptic cell. This is where communication is transferred in the nervous system. So that these little vesicles full of neurotransmitter, which fuse with the surface of this cell to release their neurotransmitter onto this cell. So this is the presynaptic cell, this is the postsynaptic cell, this is synaptic transmission. The fusion of these vesicles, release of neurotransmitter, and the activation of receptors over here. Okay. Synuclein is not just at all these places. It's specifically presynaptic. It's not in the postsynaptic cell, it's not in the space here, it's actually right here in the nerve terminal. And here just shows staining in some neurons that we put into culture where we do a lot of these experiments. And you can see this is an antibody that labels synaptic vesicles, and this is an antibody to synuclein. And they pretty much look identical, because basically synuclein labels where the vesicles are in the presynaptic process. Okay, so this is a place where the neurotransmitter is released. The brain does a lot of stuff, but what this says is synuclein does something to the release of neurotransmitter. What it is remained a mystery. Okay, so this is the big question. It's there. We all have this protein. What does it do? So, okay, a little bit of details here. So, as I mentioned before, synuclein is a small protein, about 140 amino acids, and it's got these little repeats here at one end of the protein. It's highly conserved in vertebrates. But, for example, worms and flies, they have neurons, they have synapses, they do a lot of stuff like what I just mentioned from the previous slide. They don't have any synuclein. Only invertebrates, fish, animals with spinal cords. And there are actually 3 versions of the synuclein gene, alpha, beta, and gamma. Only mutations in alpha cause Parkinson's. But there's 2 other genes that look really similar. And there are no other genes like it that we have. They're totally unique by themselves. And that's part of the mystery. It's like they're not like anything else that we have learned about before. And there are 2 things that you need to know. One is, first of all, in solution, that is, we take synuclein, we put it in solution. It's called natively unfolded. That means it's kind of flopping around in the breeze. Okay? But when it aggregates in Parkinson's disease, what it does is it forms these fibrils. And that's the aggregate. The aggregate are these fibrils. That's the so-called bad stuff that occurs in patients' brains. On the other hand, synuclein also has a different shape, which is called a helical shape, which is a coil. That's what we think of as the normal shape of the protein. So I'm showing it here in a diagram on synaptic vesicle, which is where it's thought to be located. And this is looking at the coil. We're looking at it from the side. It's lying along the surface of the synaptic vesicle as it's called, and the end of it here is sticking out in the breeze. And this is highly conserved. That means it's present across all species, alpha, beta, gamma, all have very similar sequence here. This seems to be the important part of the protein. It binds to these membranes. That's the key for what this protein does. Its interaction with the biological membranes of our cells, particularly synaptic vesicles. So that's the basic idea. This is the normal shape of the protein, this coil. This is the abnormal shape, the aggregate. And there are two things that I'm basically going to tell you about our own work in this area. And as I mentioned, there are two ways to study this. One is you take it in a test tube and you study its properties. But we've done all these experiments in cells, in particular in neurons, so that we can understand what it's doing in the place that we care about. So, and that's extremely important here. So, what we've learned, to summarize a lot of experiments, and this is what many people are quite interested in the field because they think this will influence the aggregation of protein. What we've learned is that synuclein seems to associate with these little synaptic vesicles that contain the neurotransmitter and release it upon stimulation at a synapse. And then when you stimulate, so in neuroscience, people that study neurons and the brain, they want to know what activity does. Just like you guys want to know what physical activity does. Physical activity, everything our brain does is activity of nerve cells. They're firing, and they're active release of neurotransmitter. What does activity do to all these things? So basically what activity does to synuclein, when you stimulate these neurons to release neurotransmitter, synuclein comes off the vesicle and floats away. This is a clue to its function. So it's on the vesicle here. When it fuses with the plasma membrane, it comes off and floats away. There's another protein that comes off before synuclein, and then synuclein comes off at the step of fusion. Okay, what that means is activity. I mean, if you think in very general terms, activity does something to the behavior of synuclein. Activity causes it to move from being bound to the membrane, where it might actually be a good thing in its normal state, to floating around free, where maybe it can misfold and do some bad stuff. But there's a gross oversimplification. The point is that activity does something to the protein. So that's what activity does to synuclein. The big question is, what does synuclein do? What does it do in cells? We all have this protein. Why is it there? This is the mystery. Amazingly, almost 20 years since the identification of these agents in synuclein, we still don't know what this protein normally does. So, and this is a general theme. So, tau and APP, or A-beta peptide, accumulate in Alzheimer's disease, and several other degenerative disorders. Very similar situation. We don't really know what they do. So, synuclein is similar to that. And the story is people take these proteins, tau, A-beta peptide, synuclein, they put them in a test tube and say, it does X. But the gold standard test for what a protein does, or any gene does in our body, is what happens when you take it away? What changes? That's what it's normally doing there. You can make more of it and get it to do something weird, but what is it normally doing? That's what we want to know. Very simple question. And the way you do that is you take mice and you get rid of the gene that encodes that protein. So that was done with tau. That was done with A-beta peptide. Basically, they look fine, particularly tau. Looks fine. Get rid of synuclein. The mice look fine. They're running around, they eat, they do all kinds of stuff. In fact, you can get rid of all the synuclein genes, alpha, beta, and gamma, so they don't have anything like that. So it's a mystery. It's like, there's not obviously something wrong with them when we get rid of these genes. So there was one clue a while ago, before some of the work that I'm going to tell you about what this might do. So it's a little complicated, but there's another gene which helps to maintain your nerve terminal, keeps it going. When you get rid of that gene, the nerve terminal, that part of the synapse, falls apart, degenerates. It's bad. It's not Parkinson's disease, okay? But it's a form of degeneration. And if you take those animals and you make them overexpress synuclein, you make more of it, the so-called bad thing, you have to make more synuclein, it fixes their problem. So synuclein is good for something. We take it away, don't see much, but if there's a bad one particular type of problem, when we make more of it, it fixes it. So it's bad, it's also good. Okay, one parenthetic comment. One of the major efforts in Parkinson's disease management these days is to make synuclein go away, because it's supposed to be bad. It is bad in some way, but the point is, it's good in some ways. And of course people are worried, well, if we make it go away, it won't do the good thing. They don't know what will happen. And so this is important for many reasons. But so when we started doing these experiments, we just did some simple things where we would make more synuclein to see what happens. This is like to mimic what's happening in patients that have an extra copy or two of the gene. Can we mimic and find out what that does to cells? So what we did was we would take neurons and overexpress synuclein 2 to 3-fold. That's like having an extra copy or two of the gene. And the important thing about this is the cells were not dead. There were no aggregates of synuclein. Everything looked okay. We just had a little bit more protein. Because what we're really interested in is the first thing that happens, not the last thing when they're falling apart. So that's why we did this. Just a little bit more synuclein. What happens acutely when we use this. So this is again a synapse, as I showed you before. And what we do in these experiments, the details are not important, is we're imaging the fusion of synaptic vesicles with the plasma membrane. So when activity stimulates these cells, the vesicles fuse, release their transmitter, activate receptors in the postsynaptic cell here. That's synaptic transmission. And so we're imaging the fusion of vesicles. We're directly imaging the behavior of these vesicles here. And when the fluorescence goes up here, it means the vesicles are fusing. And there are many vesicles fusing. So this is the cumulative fusion of many vesicles when we stimulate them 10 times a second for a while here. It's pretty strong stimulation, but it's what a lot of neurons do. And so this is the cumulative fusion of those vesicles, what we call exocytosis. And when we overexpress it in just a little bit, I mean, we seriously impair it. So exocytosis becomes considerably slower. That's a pretty big effect, actually. We did this in mice too, and they probably have some behavior problems, not terribly serious, but they also have a defect in synaptic transmission in an animal as well as in our cultures. So what happens here is it slows, so overexpression, making more synuclein, slows exocytosis, inhibits synaptic transmission. That doesn't sound too good, okay? So what I'm going to skip over a whole bunch of things about the way we got to what I'm going to tell you next, just except to say that we and others around the same time found that if we took synuclein in a test tube and added it to membranes, these are not normal cell membranes, these are just artificial membranes we make and put in the test tube, and we put synuclein with them, it turns the membranes into spaghetti. They're normally big sheets to begin with, and they end up as these tubules. So synuclein is basically mending the membranes, shredding them, and turning them into these tubules. So this is a clue from in vitro work and other experiments, which I don't want to get into. And so this immediately suggested some things to us about, more specifically, what synuclein was doing at the nerve terminal. And this is an image, again, a diagram of the fusion of vesicles containing neurotransmitters and peptides and other stuff. Here's a vesicle. Here it fuses with the plasma membrane, releasing the neurotransmitter. And basically what's happening, we form a little pore here. The pore dilates, it dilates further, and then the membrane collapses into the plasma membrane. It is eventually retrieved by a process called endocytosis. And sometimes this pore here, a lot of people have studied this, the pore can change its mind, close back off again, basically go backwards, and pinch the vesicle off. So this pore can control the release of transmitter. But the first thing we thought of when we saw synuclein was bending membranes is maybe it would affect the endocytosis, the recovery of these vesicles from the plasma membrane, where you have to basically turn the flat membrane into this curved thing. Maybe synuclein would do that. We found no evidence of that whatsoever. So okay, that would have been the first thing for it to do, but maybe it does something with this pore. So we specifically looked at this, and I don't want to get into all the details here, but basically we have a way to look at the fusion of the individual vesicles. We do it in cells first that were not neurons, and then we did it in cells that are neurons, and basically showed the same thing. And so these are the kind of experiments that can be done by focusing on what this protein is doing in a cell, as opposed to intestine. So what we were wondering is whether synuclein might affect the dilation of this pore, because there's a lot of membrane bending going on here. Synuclein bends membranes, maybe it does something to this. We don't really know. Let's take a look at it. So we use this one preparation where you can look at the fusion of individual vesicles. And so this is a little movie of what these look like. And we do a lot of experiments like this. These are actually cells from the adrenal gland, where they actually release catecholamines, dopamine too, but they're not neurons. We did similar experiments in neurons, but we can do more powerful experiments in these cells. And what we use is a little peptide here with it's a fluorescent protein. And when the vesicle fuses, you see a little brightening. That's because the fluorescent protein, the fluorescence is quenched inside the cell. With fusion, it's exposed to the outside and gets a lot brighter. After that, the peptide. So here, the opening of this pore, you see brightening. As the contents are released, you see the fluorescence go away. So this is what this looks like. You see these are the individual vesicles fusing. You'll start to see a whole bunch more of them soon. And here you see the individual vesicles fusing. They brighten and then they're gradually released. And basically, the properties of these, how suddenly it increases, how long it lasts, how quickly it goes down, tells us about the opening and closing of that pore. It's a very specific thing we're looking at here. So we do a lot of these, we make a lot of these cells, we image this, and we do it in 3 different conditions. One is we look at the normal cells from the adrenal gland. Another is we look at cells that are overexpressing the normal human alpha-synuclein. These are mouse cells, but we're making more human synuclein, sticking it in there. And the last is what we call the knockout. That is lacking alpha, beta, gamma synuclein. No synucleins, all gone, to see if we see anything. And basically what we found when we did this is when we overexpress synuclein to mimic what goes on in the disease, we get fewer release events. The events per cell go down. This is what we'd seen before, and I showed you before with synaptic vesicles. Synuclein overexpression inhibits the fusion of vesicles. When we take all the synucleins away, well, we don't get any more. So the key here is the normal function of the protein. If overexpression does one thing, and the loss of the protein does the opposite thing, that means the protein is doing the same thing in both cases. So in this case, we're making more synuclein, we inhibit fusion. We would expect, if that was the normal function, and we take the proteins away, we should see more fusion, because the normal one would be inhibiting. We take it away, we get more fusion. But that's not what we see. This is exactly what everybody else has always seen, that the loss of the protein doesn't do anything. Only the overexpression of the protein. So a tremendous frustration, because it doesn't tell us what the protein is normally doing, just what happens when we overexpress it. So that was the typical situation as we saw here, but we wanted to see what happened to the pore. Much more specific question. And so basically these are what an individual event, this is a fusion event. The brightening here, the increase in fluorescence, is when the fusion occurs, and then gradually the peptide is released afterwards. And we see all different kinds. We and others have seen this many times with these particular vesicles. Sometimes the pore opens and it just sits there. And then after a while it decides, okay, now it's time to release, and it releases the peptide. Here's one where, no, the pore opens and it releases, but it doesn't release everything. And here's some where it sits there and then doesn't release it. All sorts of combinations. And this is what's happening. So basically when the pore opens, it gets brighter. And then as the peptide is lost, it gets less bright. And basically if this pore changes its mind, it's opening, then it changes its mind and decides to close again, you'll basically get this, where you won't release everything. Or if it doesn't open very much to begin with, it'll just sort of sit there and won't be released because it can't fit through that pore. And then maybe eventually it will be dissolved or the pore will open and it will come out. So basically we look at all the gory details of these things, we put them all together, and the bottom line is, without getting into all the experimental details, is that when we overexpress synuclein, we get more of these. When we take it away, we get more of these other guys. Synuclein promotes what we call the dilation of the pore. It promotes its opening. When we take it away, it does exactly the opposite. The opening gets slower. This is the normal function of the protein, because the overexpression and the knockout, the loss of function, have opposite effects. The bottom line in both cases, just to be simple here, is that the protein promotes the dilation of this pore, makes it go faster. You take it away, it tends to go backwards. You make more of it, it goes forwards. Okay. We can look at whether the pores open through more direct methods. I think I will skip a little bit. And the bottom line from these experiments is that synuclein does two things. Overexpression of synuclein actually blocks the fusion of the vesicles. This first step doesn't occur very much. But, so that's the so-called bad thing. That's what happens when we make more of the protein. It's not what it does normally. What does it do normally? It accelerates these other steps. That's the normal. So it makes these go faster. So overexpression does this bad thing. That would be like in the disease. This is the normal function of the protein that we think is why we have this protein. This is the whole purpose of it. And that's the normal function. So how do we make make sense of what does this have to do with Parkinson's disease, okay? So this is the key thing here. The mutations in synuclein that cause Parkinson's disease are not just distributed all over the protein. We know that if you get rid of this protein in mice, you don't get Parkinson's disease. It's not like if we just get rid of the protein, we're going to get Parkinson's. That's not what happens. The mutations that cause Parkinson's disease are very specific. They're in this very small region here, one particular part of the protein. And so we thought, okay, there's something doing something very specific. Now, every time we had looked at these mutations before, these are the mutations that cause Parkinson's disease in patients. Every time we looked at it before, frankly, they never did anything. When we make more of this protein, it inhibits exocytosis. When we make more of these mutants that cause Parkinson's disease, we inhibit exocytosis. It's the same. The mutations were no different from the normal protein, so that didn't provide any information about what the mutations are doing. So we did the experiments anyway. Somebody asked. And this is showing, yes, when we make beta synuclein, gamma synuclein, and alpha from previous experiments, we inhibit the number of events per cell. When we look at these two Parkinson's-associated mutations, A30D, A53T, they also, like we had shown before, and others, they inhibit exocytosis. Okay, that's not too informative. But what we want to know is, what do they do to the pore? That's this normal function of the protein. These are more specific, so maybe they do something with that. So the bottom line from this experiment is, to the right means the pore is dilating more slowly, to the left means the pore is dilating faster. And you can look at the summary here. This is the distributions. When we make more beta or gamma or alpha synuclein, the pore dilates faster, moves to the left, so it opens up faster. When we make, this is the endogenous in black here, it's kind of hidden there, that's what it does normally. When we make more of these, it goes faster. We look at the mutants that cause Parkinson's disease, they don't do anything. They're dead in this assay. So this is probably the first piece of evidence about what mutations do to the function of this protein. Very surprisingly, the mutants still do the bad thing, inhibiting exocytosis when we overexpress them, but they don't do the normal thing. So basically, what's causing Parkinson's disease with these mutations is a loss of the normal function of this protein. Very surprising. Not at all what we or other people would have thought. Even people that have thought about this a lot, it's very surprising. It's as if they're still there. The protein is still there. It can aggregate, it can inhibit exocytosis, it can do all this bad stuff. We just can't do what it's supposed to do normally. And that seems to be what causes the disease. So, again, our hypothesis is this protein is there normally. We all have to do certain things for us, to protect us against certain things. When it can't do it, it triggers the disease. And that's our idea of what causes Parkinson's disease. Anything, we would say, that impairs the normal function of synuclein, which is to promote pore dilation, would trigger the disease. So the mutants still do the bad stuff, but they don't do the good stuff. So people in my lab who did this, and a few incredibly dedicated, one dedicated graduate student and postdoc, and some people who preceded them, so these are who did it, and thanks for your attention.